Okay, thanks for coming in the morning. Um, in the last two lectures, I've outlined a few puzzles in the subject of bacterial physiology, which includes bacterial growth and division. And these are these four puzzles. The puzzle of balanced growth, okay, or self-reproduction, the puzzle of exponential trajectories of chemical populations and size, the bacterial growth laws, okay, and puzzles related to cell-to-cell variation. Now, in the next two lectures, I will try to present my own understanding of some of these puzzles. So there are many models by many people, but I will be talking primarily of the work that has been done in collaboration with these PhD students at Delhi University. Parth Pratim Pandey, Pooja Sharma, Harshant Singh, and Shagun Nagpal Sethi. So first, let me describe the model that I had briefly introduced in the context of nonlinear dynamical systems, describing bacteria, describing cells. And this is a simple toy model in terms of which we can see all the phenomena, all the four different kinds of things that I talked about. This is an example of all of those kinds of puzzles. And in fact, it was the resolution of some of those puzzles in the simple model that led us to a general understanding of the puzzles. So I'm going to talk about this model concretely, but what I'm going to say about this model holds for a very general class of dynamical systems, of models of bacteria. And I will indicate what is general and what is specific. So this is a model in which there are three variables, three time-dependent variables, P, T, and R, okay? And these stand for the populations of three molecular pools in the cell. P stands for the population of all amino acids, all the 20 different kinds of amino acid lumped together, let's say. In fact, it stands for other metabolites also. Okay, so this is so it could, in some senses, represent other metabolites which are building blocks. P stands for precursors, building blocks of the macromolecules, out of which the macromolecules are going to be built. T and R are two pools of macromolecules. T stands for proteins, the population of all proteins in the cell, which are engaged in transporting food from outside the cell into the cell, and then converting it into the precursor molecules. Okay, that is the entire metabolic network of small molecules, okay, of metabolites. Okay, so all the enzymes, all the proteins which are used there, enzymes, okay, the pool of that is what we were calling T. And R stands for ribosome. Ribosome is a familiar object to I guess all of you in the cell, and the ribosome consists of RNA and protein molecules. This stands for, R stands for the pool, the number of ribosomes inside the cell. Okay. Okay. And while of course ribosome is made of both RNA and protein, we will behave as if this ribosome is constructed out of amino acids only. Okay, so it's just proteins that It's just the ribosomal protein, if you like, that we are considering. Okay? So these arrows mean that the food molecules which are outside the cell are coming into the cell and getting converted into precursors. And this green arrow means that the transporters and other metabolic enzymes are the catalysts for this coarse-grained reaction inside the cell. So, this all metabolic network has been lumped into a single reaction. Food going into precursors, and that is catalyzed by this pool of enzymes or molecules, T. The second reaction is that the precursors, amino acids, are being converted into proteins, they're being assembled, the macromolecules, the enzymes are being assembled into from the amino acids. So here, these guys are getting used up, the precursors are getting used up, and the enzymes are getting produced. And all this is also lumped into a single reaction represented by this red arrow. And this single reaction is catalyzed by the ribosome. So this green arrow represents the catalysis due to ribosome. Okay. And finally, these precursors, amino acids, etc., are being used to make the ribosomes themselves. The ribosome also makes ribosomal protein. Okay. And so that is this reaction, amino acids producing ribosomes, also catalyzed by the ribosome itself. So there are three reactions, each catalyzed by something inside the cell. And therefore, you can write down an equation for the concentration of P, okay, inside the cell, d by dt, is one term which is the production of these enzymes, right? Sorry, not of these enzymes, of these precursors, production of these precursors, and that is proportional to how much enzyme you have, right? Because these enzymes are catalyzing its production. If there are no enzymes, this is not going to be produced, okay? And so, for simplicity, it's just proportional to the number of enzymes that is present, okay? The amount of enzyme that is present, the concentration of enzyme, and this constant Kp is the rate of production of P per unit enzyme. Okay, that's what it is. Okay, and what does it depend upon? It depends upon how much food is available outside, the richness of the medium outside. Okay, it also depends upon how good the enzymes are, how fast they are as catalysts in converting the food, bringing it in and converting it into precursors. So Kp depends upon the external medium and the efficiency of your enzymes. Okay. I'll come back to this term later. The production of T depends upon the concentration of the precursors. So if there are no precursors, you won't have any enzymes being produced. Okay. So this P is a factor that appears here. And if there is no catalyst for this reaction, again, you won't have this being produced. So unless you have ribosomes, you will not produce the proteins. So it's proportional to the concentration of the ribosome and the protein and the precursor. Okay. And there is a proportionality constant. Okay. So I'll come back to the proportionality constant. Let me first tell you the dependence on the variables. The rate of increase of ribosomal concentration is also proportional to, because ribosomes are made of precursors, it's proportional to the precursor concentration. And since ribosomes are catalyzing themselves, it's also proportional to this with another proportionality constant. Okay. And what is this? This term is basically the consumption of amino acids. The precursor pool is being eaten up in these two processes. Okay. So this is simply telling us, that it's being taken up, and it's being taken up in proportion to the rate that the other things are being produced. Okay. And so what is this little k? This little k is the peptide elongation rate per unit concentration. Okay. So think of little k times the concentration of P as the rate at which ribosomes are adding amino acids, okay, to a growing protein chain. Okay. So each amino acid that they add takes away from the pool of amino acids. Each amino acid that is used to make protein takes away from the pool of free amino acids available, precursors available. So this is that rate at which they are adding amino acids. And this here, okay. So now MT and MR stand for the number of amino acid monomers in a typical enzyme and in a ribosome. Okay. So if you're eating up certain amino acids, the number of T molecules that you're going to produce is going to be proportional to that same rate K, but it's going to be diminished by how many amino acids that you need to make a single protein molecule, right? So that ratio is here. Similarly here, it's proportional to K, which is the peptide elongation rate, and inversely proportional to how many amino acids you need to make a single ribosome. That MR is that number. And FT and FR are respectively the fraction of ribosomes that are engaged in making either T or R. Right? So you have a certain pool of ribosomes. These ribosomes ultimately are sitting on different messenger RNA molecules to make the respective proteins. The fraction of them that are sitting on the messenger RNAs coding for the T proteins, that fraction is FT, and the remaining fraction, the fraction that is sitting on the messenger RNA, if you like, that are coding for the ribosomal protein, that's F. So it's the question. True. I'm ignoring the RNA. In this coarse-grained model, we are ignoring the fact that that RNA has to be separately produced through the genes. Okay, we are basically saying that all that will adjust according to the rates that are required for this process. The point is that what we are assuming is the proteins are a very significant component of the ribosome, and what we are assuming is that the RNA, the ribosomal RNA that is required for this, will be a kind of a slave process that will produce the right amount of ribosomal RNA, provided you have produced the right amount of ribosomal protein. Okay. Yes. There are three components. Yes. Yes. That is what. The third is the constitutive part. Yes. So we are ignoring that. Yeah, we've ignored that. In the simple model, you can just add that part, which is a constant fraction of everything. Okay. So there is a pool of enzymes which are neither involved in metabolism nor involved in RNA, proteins which are not involved in RNA, and which is a constant pool. You can introduce that, and that is going to be an additional sector that you can add on to this one, which will have a similar structure. Okay, and then you can see that so things will not change very much. Everything that I have to say goes through with that sector included. Okay. And so you can have that detail in, and it will not affect anything that I have to say. Okay, so it will affect obviously quantitative things, but nothing of the conceptual things that I'm talking about are affected by that. You can add that. So the models that they have considered are linear models. In their models, you don't have this non-linearity. You don't have that the rate of production of the P is proportional to pH. They have KPT minus KR, okay? And here also, this is being produced in, okay, so that non-linearity is not present in those models. Furthermore, and I will tell you other things which are not there in those models. So we'll as we go along, but at this point, I can just say that. Yes.
So Kp depends on MT and MR.
Kp?
Yeah.
No. Kp, this constant, this has to do with the nutritional sector, with the sector of how good is your medium. Okay, it's um, uh, this is uh, uh, basically if I make the medium richer, then this Kp is going to increase. And if I improve the enzymes, improve the efficiency of the enzymes, that's what it'll do. Okay, but per se, it does not depend upon these numbers. Now, if you are looking at any such model, so this is in terms of concentrations. If you're looking at it in terms of populations, if you're looking at it, sorry, this is a generic way of writing these things. These equations are written in a fixed volume. Okay, if you write, if the volume of the container is fixed in which this is happening, then you will have equations like this. But since we want to talk about a growing cell, we have an expanding volume, and so we must add these terms to each of these equations. Okay, this term to each of these equations, which is the dilution, the so-called dilution term. The population. So there's a negative term in each of these equations, which takes into account the fact that every concentration, just by virtue of the expanding container, is diluting, is becoming smaller. And so that's a V dot by V term, where V is the volume of the container. And so, I will use this notation interchangeably. Xi and Ptr. So X1 will be P, X2 will be T, and X3 will be R. So, yeah. Now, okay, let us write these equations in terms of the populations of Pt and R. Okay, the actual populations, which are the variables in terms of which I have described earlier questions to you. Okay, so here you have box brackets. These represent concentrations, and Pt and R without the box bracket around them will represent populations, and they are related by simply, the populations divided by the volume is the concentration. That's so now, you can write down P or T, if you like. T is volume into the concentration of T, right? And therefore, if you want to write an equation for T dot, capital T dot, then it's concentration T dot, and you can also write down V dot. Okay, so you can write down that equation, and that is very simple. So that gives you the following structure. So if you look at T dot, it will contain two terms, because T is V times the concentration of T. So you'll have two terms. And what is going to happen as a consequence of this extra term is that the dilution term that you had in your concentrations will exactly cancel. So this sort of typically always happens. So when you write the equations in terms of the populations, you don't have to write the dilution term. Okay, the dilution term is in a sense already included. Okay, if you like, this volume is now time dependent. This volume is time dependent. Okay, so now this is a simple model. It's a nonlinear model, and as in typical nonlinear models, you don't get this phenomenon of balanced growth. Okay, you don't get the phenomenon of concentrations, doubling together and so on. You can simulate this model, and you will find that kind of a behavior. Okay. Notice that this model has not been completely specified. Supposing I know all the rate constants, all the parameters that are there. Supposing I have told you what their values are, I still can't give this model to the computer and solve this differential equations. And the reason is that I have not specified what is this volume. Okay, so the model has not been completely specified, and you can see this here in the population version of the model. But if you go back to the concentration version of the model, The concentration of the model version of the model is here. And even this is not completely specified because I have not told you what V dot by V is. Okay. Even if I know all the constants, I can't give it to the computer to simulate this until I have made a commitment as to what is V dot by V. Okay. So we need to do something about that. And the way people typically solve these models is either by assuming V to be a constant, okay, which is, they're thinking of just a constant volume container, or they assume that V dot by V is a constant equal to mu. So it is assumed usually that V dot by V is mu, and this is an externally specified constant. When you do that, then you get another parameter in these equations. Okay, you get another parameter in these equations, which is this externally specified constant mu. Okay. Alternatively, what these equations mean, if V dot by V is equal to mu, and if this is a constant, this means this is tantamount to saying that V of t is some constant V naught into e to the mu t. This assumption and this assumption are exactly the same. Okay, you can derive one from the other. Okay, so that tantamounts to declaring that this V is given by exactly this function. So these are the assumptions, and this is how, for example, Terence et al. Also solve their models. Yes. So we will apply it to the exponential phase. Okay, it can be used in any circumstance, and this model is general. And we will see that this model addresses transients which take you to the exponential phase, for example. Okay, this model in principle is valid even there. So okay, whereas when other mathematical models of cells, because of this assumption, there is an implicit assumption of exponential growth put inside those models. This is one of the things that we want to understand from first principles. We want this to come out from the models rather than being put in. Okay. So therefore, we are not going to do this. Okay? And this is the major departure of the models that I'm going to talk about with respect to other models that exist. So what are we going to do? We know that the cell is expanding. We would like to make, and we know that the volume of the cell depends upon what it contains, what is happening inside the cell. Okay. So for example, if molecules are entering the cell through some method, then osmosis will also cause water to enter into the cell. Okay, and that's what will increase the volume of the cell. So the volume of the cell we would like to take to the level of a dynamical variable. Okay, we would like to make V is equal to V of X, or in this case V of P T R. Okay, that's what you would like to make. So that the thing is endogenous, and what we do, okay, is make this assumption that V is itself a linear function of the populations that it contains. Okay, so in general, we make the assumption that V is equal to I summation Vi Xi, I going from 1 to P. Some of these Vi's might be 0, some might be non-zero, etc. So, but anyway, so it's some linear function of the X. Actually, we make a slightly more general assumption than this, but for the moment, let's just stick to this. And in particular, I will show you the example in which we say that capital V is just little v into P plus T plus R, just the summation of everything that's inside. Okay, and this is something that is motivated from osmosis, from the biophysics of osmosis. Okay. Now, the moment you do that, This is a new parameter that you're introducing into the problem. This little v is a new parameter that is entering the problem. But your volume is a dynamical variable now. And well, if the volume is given by this expression, you can ask what is v dot by v? After all, so v dot by v will be little v by capital V and plus p dot plus t dot plus r dot. From the definition, it's going to be that. Is that fine? And p dot, t dot, r dot, you've already made assumptions in the model as to what they are. So you plug those in, and this is what you get. Okay, you get this. Okay, and yeah, okay, so that's what you get. And so you see that the growth rate is now something which has become endogenous in the problem. It's not something which is exogenously specified, but you have something which depends upon your dynamics. The growth rate now depends upon the dynamics, okay, which it should, okay, and there is a new parameter that is also entered. Yes. Yes, that is the concentration. So you see that there is a factor of volume here, and there is another factor of volume which is coming from here. So what you will get is v squared in the denominator. And when you have p divided by v, that will become the concentration of p. And when you have r divided by v, it will become the concentration of r. Right? So notice that the whole structure is such that the concentration is a function of the growth rate is not a function of populations. It's a function of the concentrations only. Okay, so that's what turns out as a consequence of this linearity assumption. This is something that turns out. Okay, and now you see, of course, once you have specified what v is, it's given by this. Then notice that has introduced additional nonlinearity into the problem, right? There were these xi dependencies that you already had, but now you have additional xi dependencies in the problem, okay? And so that's your model. That's your model now. And but your model is now completely specified. You need to specify this parameter v. Once you specified this and all other parameters, then you have a set of equations that you can give to the computer and solve them for any initial condition. Is this point clear? Not only is this model completely defined, but so is the concentration model completely defined. Sorry. So, v dot by v is just mu. Okay. And now you have an expression for mu in terms of concentrations, right? So you can plug in that expression for mu in terms of concentrations here. And now you have a new model here in which the concentrations are appearing not only in these terms, in the original terms, but they are also appearing in a nonlinear way in this dilution term. Okay. So notice that at the level of concentration, there are new dynamical terms which have entered the equation. This assumption of making the volume dynamical has this consequence. Okay. So that's what we do. Now, we then simulated this model with these assumptions. Now you can give it to the computer and simulate. And so I will, for some values of parameters, we simulated the population. So let's look at these two curves. These are with two different initial conditions, but the same parameter values. So all the parameters, K, Kp, Mt, Mr, etc. You give them some numbers, V, little v, and so on. You give them some numbers, and you start with some initial condition which is at t is equal to 0 here, and you let those three differential equations run, and you see that there is a transient, and after that transient, it gets into this straight line phase on this semi-log plot. The populations are plotted on the y-axis on a logarithmic plot, and time is on a linear plot. And so you see, so P is the green one, okay, this is T and this is R. And so you can see they end up growing exponentially. The straight line means that they are growing exponentially. And the fact that these straight lines are parallel means that they are all growing exponentially at the same rate. Okay, so that's what happens. If you do it for any other initial condition, you have a different kind of a transient, but they all end up in exactly the same growth rate, okay, with the same relative ratios as before. Okay, so that's what happens, and you see this exponential growth. Okay, because they are in this state, they are all growing exponentially. So you know what they are in this phase. In once not in the transient, but in this region, we can model them as xi is equal to xi 0, some 0. Let's say I start here and I call that t is equal to 0. Okay, that time e to the mu t for some mu. Okay, and I will call this mu star. Okay? This is some constant mu which has emerged from the dynamics. So you are getting a solution which is an exponential solution which you expect for linear systems. Okay, that is what is coming out at long times. Yes. Exactly. So this is just, you know, miraculously almost coming out of yes. Yes. So I'll show you that you can actually substitute this ansatz. Okay, P of t is equal to P of 0. Let me call it P naught e to the mu star t. T of t is equal to T naught e to, and R of t is equal to R naught e to the mu star t. You can substitute this in the dynamical equation of the model, and this is a solution. This ansatz is a solution, but it is only a solution if mu star is a certain fixed function of the parameters, and these P naught, T naught, and R naught bear certain very definite ratios with respect to each other. Okay, so you would find that this ansatz is a solution, but it imposes constraints on the growth rate and on the initial concentrations. Okay, and so I will come back to that this is precisely, it turns out that this is a solution, but your motion, your initial conditions have to be along a line in your three-dimensional phase space. Yeah.
What you have provided, it is not any approximation.
No, it's not an approximation.
Maybe we are solving for last time approximation. That's why we are doing. Oh, this is what you are doing? Nothing like that.
No.
You are doing.
Thank you for that comment. Thank you. Yes. So this is an exact solution. This is an exact solution of the dynamics. And what these curves are suggesting is that this solution is an attractor of the dynamics. That is, you can start from any initial condition and you will eventually converge to this solution. Okay, that's what it is telling us, which itself happens to be an exact solution. Okay.
So is there any specific constant that determines that the population of R at a large time scale will be lower than T and then P? Like
yes,
if we can
yes,
see
it turns out. I mean, so we have, I mean, these simulations, of course, have been done with a specific choice of parameter values. For that particular choice of parameter values, it turns out that it is like this, in which R is smaller than this. And those parameter values have been chosen with an eye on what we know those parameters to be experimentally. Okay, so but it does not matter what those parameters values you choose. For the moment, what I want to convey is, doesn't matter what parameter values you choose, doesn't matter what initial conditions you choose, this feature that after a transient, things will enter into an exponential growth with all the populations growing with the same exponential growth is a feature all the time for this model. Okay, this slope will depend upon the parameters, But the fact that you end up in an exponentially growing trajectory is not, doesn't matter what parameter values you choose. This model will always give you that. Yes. There was a question? Yeah. K, little k. You can. So the parameter k has to be exogenously specified. And k is the rate at which a single ribosome will add amino acids will eat up amino acids, okay, per unit concentration of P. Okay, so that's the interpretation of K, and that's a parameter that you have to put in by hand. That's not something that we are explaining. Okay, we are explaining other features of the dynamics, but that's something that we have to put in by hand. No. Yeah. Yes. Can you? Yeah. Yes. V of T. Yes. We are now. So all these are now models of a single cell. Okay. We are now talking of the dynamical system that describes a single cell, and it is described by the three populations that it contains. We'll come to that. Okay. So this is pure growth. Okay. And it is exponentially growing. The volume is exponentially growing from this point onwards. In this period, it's not exponentially growing. Why? Because you can see that R is growing at some rate, P is going at some rate, T is growing at some other rate. So volume is a sum of P, T, and R. Each one of them is growing at their own respective rates. So the volume is not growing exponentially in the transient. It's only growing exponentially when all these variables have started behaving this ansatz, according to this ansatz. Right? When all of them have started growing exponentially with the same rate, then V is also growing exponentially with the same rate. Right? Is that clear? Yes. And a consequence of that fact is that because V is growing at the same rate as each of the populations, it means that your concentrations have actually become constant in time. Okay? This attractor for the populations, along with the fact that V is a linear function of the populations, means that V and each population increase by the same growth factor, right? Because if I just substitute here, Xi is given by this e to the power of mu star T will come outside, right? And that's the only time dependence that the volume will have. And so the volume is also growing exponentially with the same factor. So therefore, the concentrations have become constant. And if you look at the concentration equations, you find that it converges to a fixed point attractor. Okay, so the population equations converge to this attractor. The concentrations converge to a fixed point attractor. Is this point clear? That's so the fixed point means that all the concentrations have become constant in time. So if you have a dynamical system in which you have variables x1, x2, x3, and let's say these x1, x2, x3 are the concentrations of P, T, and R, okay, then a fixed point is one in which the right-hand side, so little xi dot, okay, is equal to some functions on the right-hand side. They've all become 0. Okay, so you have a fixed point attractor. That's what's happening. Exactly. All the K's, yes. I will give you the formula. Okay, so you can exactly solve for it. Okay, and you can explicitly write down an expression for mu, and that in fact is what we use to understand the bacterial growth laws. Okay, there are relationships, okay, between these variables. You see these P, T, and R. I mean, okay, these constants, the initial conditions, the ratios between of the populations and the growth rate. And that becomes the origin of the growth laws of bacterial dynamics. Okay, so so I will come back to that. Notice also that because because all of them are increasing with the same mu, their ratios have become constant. So P of T divided by R of T is equal to just P naught by R naught, and that is constant in this regime. In this regime, the ratios are constant. Now here, what is actually plotted is the ratio T by R. Okay, so if this was the right description of these curves, then the ratios should have become constant, and they indeed do. And here you can see the set of initial conditions for which it has been tested ranges over 20 orders of magnitude. Okay, it doesn't matter what initial conditions you start with, they will all converge to the same fixed point. Okay, so okay, these are different initial conditions and so on. So now it turned out that so this happened. And we just thought, I mean, okay, so this is happening, and we were, we noted the fact that there is an exponential growth in this model, but we didn't realize that this is actually a general phenomenon that will happen when you make the volume dynamical in the manner that we have done. Okay, and so we were trying a complicated model of the cell in which there are 30 to 40 dynamical variables. Okay. I had mentioned this in my earlier talk, and I mean, there is an external food, there is an internal food, there are internal metabolites, there is ADP, there is ATP, there are enzymes of various kinds. Okay, there is the RNA polymerase is there. So many cellular variables, and you have these reactions in which these variables are being produced, these chemical species are being produced and consumed. So you have these nonlinear differential equations, and these nonlinear differential equations have nonlinear terms inside. That is V2, V3, nu2, etc. These have cubic terms inside them, and so on. So this is even more nonlinear and much more complicated and decentralized model. Okay, but again, we assume that the volume is a linear function, the sum in this case again of all the populations. And when you do that, and we simulated this model, and again, the same kind of thing was happening, that after a transient, it was ending up into an exponentially growing, all the trajectories were growing exponentially, and they became parallel, so that means they were growing at the same rate. Yeah. And the ratios were becoming constant in time, independent of initial conditions. Okay, so that is a feature that surprised us very much, that why is this happening? We didn't expect exponential growth, and that seemed to be happening. And then, so this is the answer to why we are getting exponential growth. Okay, so now our understanding is the following: that the functions Fi are highly nonlinear, but they are nonlinear in a very special way. Okay, they are nonlinear. So now, so our dynamical system is xi dot is Fi of x. Okay, and these capital X's, okay, these capital X's will denote the populations, as I said. So so that's what it is. And for the PTR model, x1 dot, which is p dot, is given by this function kpt minus. Okay, that's my f1. This is my function f2, which is for t dot, and this is my function f3 for r dot. Right? These are the same functions that I wrote down in the previous. This is this particular model. This particular model. I will make a result for general models in a moment. Okay, for all models in a moment. So, but let's look at this and let us notice the following feature of this model, which is responsible for giving us exponential trajectories. Okay, you see, this volume has now become a function of Pt and R, right? This volume is now a function of Pt and R, and it is in fact a linear function of Pt and R. So these functions satisfy the following the following scaling property. That is, if I scale all my PTR, all the variables xi, by some constant beta, okay? So beta is some non-negative, some positive number. I scale all of them by beta, okay? Then my function itself gets scaled by beta, okay? So let's see that for this one, right? I mean, if we take that here, so let's see. So clearly, this is a linear function. If I take this term, just kpt, if I take this term, that certainly satisfies this property. That if I multiply all the variables by beta, then that term gets multiplied by beta because it's linear in t. Okay, but this is a quadratic term in t in my variables. But v is linear, and therefore this also has the same property that we have chosen. That's our choice. Exactly. Okay. So our choice of v as a linear function of the populations is converting this nonlinear function. It remains nonlinear, but it is nonlinear in this with this very special property that it is a homogeneous degree 1 function of its arguments. Is this clear? That PR by V will have this property. So, right? Should I just write it down? So let's take this PR by V. This is equal to PR by little v into P plus T plus R. Right? So this is, let me call this function f of PTR. Now, let me consider f of beta P, beta T, beta R. This is going to be beta P, beta R, V into beta P plus beta P plus beta R. And what is this? You see, this is a beta which will come out of this one. Okay, this is going to be the following. This is going to be beta into PR over V, P plus T plus R, which is just beta times F of PTR. Okay, that's all I'm saying. Is this clear? So this property is arising because in this quadratic term, there is 1 power of V sitting below. And in this other model, in this more complicated model, you had a cubic term and that came with a V squared sitting below. Okay? So that's the reason why this model also satisfied the same property. Okay? So because of this property, you can immediately see that this ansatz is going to be an exact solution of the model. Okay? Why? Because let's substitute this ansatz into this equation. This is my original model. Let's substitute this ansatz in which xi of t is xi of 0 into e to the mu t, where this mu is the same for all I. Okay, the same mu for all I. Okay. Let's substitute this in this, this equation, this ansatz into this, and see whether it's going to be a solution or not. Okay, let's check, and you can do that. So, if this is your ansatz, then dxi by dt, you can immediately do here. That is simply mu times this whole thing, mu times xi of t. The left hand side is just mu times xi of t. And the right hand side, of course, you have to put in each one of these terms. Each x1 of 0 e to the mu t, x2 of 0 e to the mu t, etc. Okay? So so the right hand side is this. But notice that this is as if your beta was e to the mu t. all of these x's are being multiplied by the same scale factor, which is beta. And so, by virtue of this property, this e to the mu t is going to come out, right? It's going to come out because this function has that homogeneous degree 1 property. And so, you find that your right hand side, the only time dependence it has is e to the mu t, and after that, you just have these initial conditions, this function fi of the initial conditions. And this e to the mu t will cancel this e to the mu t. So the time dependence completely disappears. And you get this equation, mu xi of 0, this is gone, is equal to fi of these. Okay. Is this clear? That whenever you have a homogeneous degree, a set of functions which is homogeneous degree 1, they will always admit an ansatz like this as an exact solution. And what this is telling you is that this will be an exact solution provided your mu and your xi of 0, they satisfy these n equations. Okay, it's not that you are guaranteed a solution. You are guaranteed a solution provided this condition turns out to be true. Okay, so that's the conclusion. And what we can now do, but this condition is time independent. This is a set of algebraic equations. Okay, and okay, yeah. This is a set of algebraic equations whose solution I will tell you for the PTR model. Yeah. Okay. So, now this sort of tells us that our systems are nonlinear. The system, after you make this change, the system is nonlinear, but it is nonlinear in a manner that is very special, homogeneous degree 1, and it shares this property with linear systems. So linear systems are, of course, also homogeneous degree 1, but our nonlinear systems have also become homogeneous degree 1. And we can ask, what exactly is the comparison between the linear systems and the nonlinear systems that we are talking about? Okay. So notice that in the case of linear systems, you have xi dot is equal to aij xj, right? And this last equation that I showed you, which the mu and the xi zeros must satisfy, that equation now starts looking like this equation. Because mu xi of 0 is equal to fi of the x's at 0. That was the condition. And look at this equation. What is this equation? This equation is nothing but the eigenvalue equation of the matrix A. Right? A linear system is defined by a constant matrix A. And in that case, the equation that we have derived for general homogeneous degree 1 systems, this equation, which we have derived for general nonlinear but homogeneous degree 1 solutions, this equation actually reduces for linear systems to nothing but the eigenvalue equation of the matrix of the linear problem, where mu is an eigenvalue, is the eigenvalue of A, and these vectors must be the eigenvector. Okay, and so what we have done is that we have found a nonlinear generalization of the eigenvalue equation of a matrix. Okay. And that is what describes our solutions, because these functions fi are in general nonlinear for us. Okay, so we have found that generalization, and just like for these eigenvectors, if you have an eigenvalue equation, you can determine the eigenvector only up to a multiplicative constant, right? The eigenvectors are only known up to a multiplicative constant. And so similarly, in this case also, our initial conditions, xi of 0, are only known to a multiplicative constant. What that means is that this eigenvalue equation actually defines a line in our phase space. Okay, so the ratios are constant, which means that what we've defined, the ratios of the coordinates are constant, right? So the condition for this to be a solution is that the ratios of the x's become some constants which depend upon parameters. And what that means is that in your n-dimensional phase space, you have defined a line, okay, which is the solution, where your system is naturally converging, okay, and when it is converging there, everything is growing exponentially. So the problem of exponential growth and balanced growth are both simultaneously solved. So you might ask that supposing I take the same model and I make a different ansatz for V. We've made an ansatz in which V is a linear function of the populations. But supposing we make an ansatz in which V is not a linear function, but let's say it's t to the 2/3. Okay, supposing we just make an ansatz which departs from that linearity assumption. Okay. Then, if V is no longer a linear function of the populations, then I will lose this property. I will lose this homogeneous degree 1 property. And if I lose this homogeneous degree 1 property, I will lose the fact that I can have a solution of this kind. Okay? And if you simulate such systems, you can take the exact same system. I've also added 2 additional terms here for the decay rate of the enzymes and the ribosomes. Okay, it doesn't matter. That's just an additional complication. Even if you didn't have that, it would be the same thing. That if you do anything like that, the moment you depart from linearity, you find that you depart from exponentiality. The chemical solutions. So here is the populations are now growing like this. They are not converging to straight lines which are parallel. Okay, and if you look at the ratios of the chemicals, they are not converging to a constant value, but they are continuously changing. Okay, so you lose exponential growth and you lose balanced growth. Okay? So balanced growth is a consequence of the special assumption that we've made. Okay, that's right. That's right. Yes. Plus y, which is also inside the cell. Oh yes, sure. So if your y is also a dynamical variable, and so then y will also be a dynamical variable in the model, and y will satisfy some differential equation based on which reactions it is being produced in, which reactions it's being consumed in, and those equations will also have this property. So indeed, that's absolutely correct. That as long as it's this result is true as long as this is true. Some of these VIs may be 0, in which case some variables are not appearing in your volume. But whichever variables are appearing in your volume, as long as they appear linearly, and they don't have to appear with equal coefficients like they do here, P plus T plus R, they can appear with unequal coefficients. Doesn't matter. As long as this is true, this scaling property will be true. Right? Is that? So maybe we should talk about it on offline. I would like to know an example like that. So all I'm saying is that in principle, if there are other chemicals involved, then those chemicals will have their own, those will also have to be your dynamical variables. And if they are dynamical variables if they are chemicals, and we are talking of a well-stirred chemotap kind of situation, then you will end up with a system of of this kind, okay, a nonlinear set of differential equations, and those so now here is a slide where I'm arguing why does it have to be homogeneous degree 1, right? If your V is linear, I'm now saying that irrespective of any chemical system, if you have any well-stirred chemical system, you will always end up at the population level with homogeneous degree 1 functions, okay? And that will also answer Nivedita's question of what is the more general class of V's that is permitted for you to have this property, this homogeneous degree 1 property, okay? So it might partially address the thing, but I think you may have something very specific in mind about some particular kind of chemical, and I'll be happy to discuss with you the kind of equations that one might write down for those, even if one doesn't know it exactly, but that those equations will satisfy this kind of property is what we can see, right? And discuss that, okay? So now, this is a feature of chemical dynamics, that the concentration, so now my notation has slightly changed for convenience. Earlier, I was writing capital Xi with a box bracket for concentrations. Now, for brevity, I will just denote it by small xi, okay? So notice that capital Xi will stand for populations and little xi will stand for concentrations, okay? So capital Xi, the populations are extensive variables, and little xi, because they are divided by V, they are intensive variables, okay? So there's a distinction between them. And all chemical dynamics that we know, all well-stirred chemical dynamics that we know, is always written in this form, that the concentration, the rate of change of concentration, the local rate of change of concentration is given by some function of the concentrations themselves, okay? So this is what I'm going to assume, that the change of concentrations, the rate of change of concentration of any chemical is proportional to the concentration of other chemicals there, which is what determines how fast it is produced or how fast it is consumed, okay? So if you accept this, my claim is that if this is your chemical dynamics, then it follows that when you write the population dynamics of systems in expanding containers whose volume is linearly dependent on the populations, and I will tell you what generalization thereof is permitted, then you will always end up for the population dynamics homogeneous degree 1 functions, okay? So let's see the argument. So if this is your population, if this is your concentration dynamics locally, and this is the concentration dynamics in a fixed volume, then in an expanding volume, this is what your concentration dynamics will be. Okay? These functions gi are now completely arbitrary. Okay, you can have mass action kinetics of any order. You can have Michaelis-Menten kinetics, as long as the concentration depends. The rate of change of concentration depends upon other concentrations. You will have some particular functional form G. Okay, you can have a regulation, you can have any kind of thing. Okay, so we have this. In the expanding volume case, we have to add this dilution term, and now we can write down capital Xi dot, the rate of change of the population, and as I said earlier, these two terms cancel. Okay, and you are left with capital Xi dot will be simply the volume times Gi of X. Okay, and that's generic. Now, let V depend upon Xi in some fashion. Okay, and we'll see what is allowed. Let us elevate V to a dynamical variable, and it depends upon the populations. And let us now look at this set of equations. Okay, what does this, what happens when V becomes dynamical? Then we can write down this equation. This V becomes a function of the capital X's. And this little x, of course, is just capital X by V of X, right? So this, we can, this combination is what we call little Fi of capital X. So we find that the dynamics of the if the dynamics of the concentrations is this, then the dynamics of the populations is this, capital Xi dot is Fi of X, where this Fi of X is given in terms of the G and the V in this fashion. Okay? That just is general, completely general. Now let us ask whether these functions F are homogeneous degree 1 or not, or under what circumstances will they be homogeneous degree 1, right? So let's substitute Fi of beta of X, and since Fi is given by this, I have a beta coming here, I have a beta coming here, and I have a beta coming here. Okay. So now, this is the more general requirement, okay? That if V itself is a homogeneous degree 1 function of the Xi, and linear is just a special case of that. But as long as V itself has this property, if V has this property, that if V of beta X is equal to beta times V of X, okay? Even V itself could be a nonlinear function of the X's, but as long as it is homogeneous degree 1, then you see what happens is that this factor beta comes out, and this beta cancel. Here again, this beta comes out, and so you have the property that, okay, Fi of beta X, which is what you were looking at, is beta times Fi of X. So as long as V itself is a homogeneous degree function of the populations, you are guaranteed, doesn't matter what your chemical dynamics is, okay? In an expanding container where V, if it happens to be of that kind, then Fi will be a homogeneous degree function X. And then such an ansatz is in principle an exact solution of the model. Doesn't matter how complicated, you may have 10,000 variables, okay? This is going to be an exact solution of the model, provided that generalized eigenvalue equations you can find physical solutions of, solutions in which all the xi's are positive, right, or non-negative, right? So you need to find solutions of that, and you need to find, okay? So that's basically the conclusion. And the illustration that I told you, when V was to the 2/3, okay? So again, your functions F had a scaling property, okay? You could construct them at least to have a scaling property, but you still don't get this one. It's only this particular scaling property that gives you this answer, no? That these exponential solutions. Yeah. There's a lot to do. There is a lot to be done, because what is being done here is setting out what I think is the proper framework for thinking about bacteria. Not only to think about bacteria, but also to think about primitive things like protocells. Okay? But we've, now we have to take models of this kind and see what they actually do to understand bacteria. I have solved the first two problems of balanced growth and exponential growth, and I've told you why, when and why those two properties will arise. But I have not shown you that growth laws arise. I have not shown you that when bacteria divide, they have the adder property, right? I mean that it's as if, what is the source of stochasticity, and how does that stochasticity organize itself in such a way that you get precisely the distributions of all the quantities that you get? Those are questions that we don't have an answer. So this answers a broad set of questions. And it seems to me that this is the framework in which bacterial cells should be modeled, in which I think I will concentrations. Here is a slide whose title I was not able to finish. Okay, and I forgot that I have not finished the title of this slide. So, but anyway, the slide itself is there. Okay. What I wanted to again emphasize, I think I've already done it though. I think that's, yeah. So I should tell you that if you have this as your definition of V, if you take it, this is not the most general form that is allowed, but this is the linear version, the simple version. Then it turns out that V dot by V is just given by this very simple function of the GI. So you have a very simple formula for the instantaneous growth rate as a dynamical variable. The growth rate is expressed as a function of the concentrations. Okay, if you know those original functions GI, those chemical dynamics, then you can immediately write down what that is. And yeah, I think I will. So this also tells us that as you reach exponential trajectories in the populations, you are reaching a fixed point attractor. Okay, in the concentrations. Okay, so I think I've belabored the point now. I think I should stop. And this is what I have, I think, covered so far. I have not covered the growth laws, and I have not covered the division stuff, and I will try to do justice to it next time. But that's what it is. I don't know if I have clearly explained this geometric picture in phase space. Okay, what has happened? That we have a line of balanced growth. Okay, the important thing that I want to emphasize is that we have not had to assume any regulatory mechanism to get us into exponential growth and balanced growth. These were just, I mean, some chemical was getting consumed, some other chemicals were getting produced. There's nothing regulation, there's no regulation about it. So this is without requiring any specific machinery, we are able to address the question of balanced growth. And there is a new mathematical structure that appears from this, which is a generalization of the eigenvalue equation of a matrix, a nonlinear generalization. Mathematicians that I have talked to have not encountered this structure. They don't know if they have actually studied these kinds of things before. So I have not been able to get anything. So there might be an interesting mathematical direction to pursue, a purely mathematical direction to pursue with this kind of mathematical structure. Here, which I will cover next time, we will turn to growth laws. Okay, so I think I'll just stop here.